New window. All right, we are live. As I said, pretty sad story. Parasite in the news. Parents reach $250,000 settlement in Texas City after son 3 dies after contracting brain-eating amoeba at a splash pad. What's the name of this amoeba? Causes primary amoebic meningoencephalitis, or PAM. Don't make me look to see who's presenting it, because someone's presenting it. It's Naegleria fowleri. Somebody's presenting it. Normally it pops up in the news every year. This is one of those that you don't want to get. Child was at a splash pad. That's at least where they think they picked it up. Kind of like it. I mean, the water's supposed to be chlorinated. I'd have to really check to see if chlorinated water is protective, because several years back, a individual in a church youth group was at a whitewater rafting park in North Carolina, man-made park in North Carolina. Out of like the 14 students that traveled down there, she was the only one that got it, and she died from it. That was also chlorinated water, presumably chlorinated water. So it's a kind of big question mark. But out of all the kids, I'm going to make this known again, out of all the kids that were there at the splash pad and that visit the splash pad, this isn't a one-time deal where it just suddenly appeared and then it's only there the one time. It's probably there, just like Nasworthy. It's probably out there. And you think about all the individuals that swim out there and all the individuals that get any sort of water splashed up into their nose whatsoever, to have a handful of people ever get it. I mean, you might not, in that one water body, you might have one individual over like 20 years ever get it. And the big question is why? Why don't more people get it? So that's probably genetics. You know, what's the medicine? Genetic medicine? No. It's where they actually look at your genetic code and try to treat you based on that. So like we talked about, with the nematodes, pretty certain we talked about it. Your race can affect your susceptibility to parasites. Same idea. Maybe there's something there where those individuals just have a specific receptor, might not contribute anything, might not benefit them in any other way, but that is the receptor that Naegleria latches onto, penetrates into the cell, travels up to the brain, and then ultimately kills you. So yeah, the chance of you getting it is really, really low, but if you get it, chance of survival is really, really low. There's only been a couple people that have survived. So, parasite in the news, kind of depressing, but thought I'd point it out. All right. So where we left off, class Kinetoplasta. So we're in our protozoans. These are could be intracellular or extracellular parasites. All right. Difficulty of this section is all of the terminology. It seems like every group has their own name for the various life cycle stages. So if we go back to your platyhelminths, we had larval stage names for all of the digenetic trematodes, and we had, I think, 5 of them, or 6 of them that we covered. Hey, no big deal. You know the names, they applied to 6 of them. 6 of those different life cycles. The cestodes had, same basic names. Started getting into some complexity because of the different metacestode terms. But for the most part, they all had some sort of oncosphere penetrating into that first intermediate host, or penetrating into the hemocoel of that first intermediate host, and so forth. And then the nematodes were fairly simple, J1s through the J4s. Some of them had microflaria. But on our protozoans, it's a terminology. So all that I could say is when we go through this, when you're studying, I would probably put together a word list and work on those terms as we go. All right, so we're going to start with the class Kinetoplasta. Members of this group can cause some pretty significant pathology, could cause some gruesome pathology, and ultimately can kill you. It depends on what parasite, what species you have, is it treated or untreated, and so forth. Members of this group will have 1 or 2 flagella, so we're in our flagellates again. All right, and typically they have a paraxial rod. This paraxial rod is an electron-dense structure. Shows up on our transmission electron microscopy. So here's our 9+3 microtubule arrangement. That's our axoneme, and then you have the paraxial rod, which is an electron-dense region. Looks like it's fibril lattice. It often occurs along the entire length of the flagella and runs and is contained within that flagellar membrane. What is it? Maybe it's some sort of structural process. I mean, if it's made up of this fibrillar lattice, you would assume that it's some sort of structure that maybe helps keep the axoneme fairly straight. You know, so it's not just like super floppy, whatever. But you're going to see this thing. Can we see it in any of our light microscopy? No, unfortunately, no. So at some stage in their life cycle, they will possess a single mitochondria. So in the entire organism, you will have this mitochondria. This mitochondria, as we know, has its own DNA. For these kinetoplasts, it could contain up to 20% of the cell's total DNA structure. And that structure is called the kinetoplast, the electron-dense area. So I believe that's on our next slide. Yep. So when you're ready. And that's why it's called this. Mitochondria is usually fairly large. The DNA-containing region is concentrated. It's called the kinetoplast. That kinetoplast is usually very closely associated with the kinetosome. So the kinetosome is, what is a kinetosome? Instead of giving you the definition, what is it? What is it? Nope. Now, our kinetosome was associated with our flagella. What was the other name for it? That's probably the name that you learned in the basal body. Yeah, it's the basal body is what gives rise to our flagella, to the axoneme. That's the kinetosome. It gives rise to the axoneme. All right, so our kinetoplast is going to be closely associated with it. These are two different terms. All right, the kinetoplast is in our mitochondria. The kinetosome is at that flagellar pocket, the base of the flagellar pocket. All right, so the kinetoplast is the DNA-containing region, and for us, it's always going to be located near the base of the flagella. It's associated with it in some way. It has a lot of interesting things associated with it that's beyond the scope of this class. So if you're interested in what's there, take a look. It's the functions. We'll mention some of the functions, I believe, a little bit later. All right. These organisms, the kinetoplasts, also contain a unique organelle called the glycosome. All right, so we have our mitochondria, right? But then they also have this other membrane-bound organelle called the glycosome. This is where glycolysis is going to be occurring. So this organelle is going to have basically all the enzymes and stuff associated with glycolysis. Why is that? What do you think its primary ATP source will be? Anybody? What is glycolysis?
First step of photosynthesis.
So what?
Isn't it a step of photosynthesis? Like the first. That's something to do. No, it's like a respiration.
Oh, it's it. Good, good. I was gonna say, did you have botany yet?
No, I had cell bio and we were together and
Okay.
it's confusing.
Good. Yes. First step. So what does it break down?
Glucose.
Glucose, right? First step. What happens after glycolysis? Yeah, then you get transferred to the electron transport chain, right? All right. Well, if we're not going to have oxidative phosphorylation, that electron transport chain, then all you're going to have is glycolysis. So this could be a reason why we have this glycosome, is that these organisms, some of these organisms, will rely on glycolysis for all of their ATP production. And we'll talk about one specifically, and we'll talk about why that is the case. So we have this glycosome. So I've got two different diagrams. These are just great diagrams. Unfortunately, these guys are super small. When you look at them at 100x, at the 100x objective, they are going to be even smaller. So kind of prepare for, diagrams like this and pay attention to what's on our word list for lab. All right, so that's our class. We're going to start with the order Trypanosomatida. The Trypanosomes. Members of this group have a huge economic and medical impact. They're going to infect nearly all vertebrates. We're going to focus primarily on those that infect the humans. All right. So what kind of diseases do they cause? You've probably heard some of them. African sleeping sickness. That's caused by the African trypanosome. Chagas disease. That's American trypanosome. And then leishmaniasis. That's the one where they were using the CRISPR technology. Leishmaniasis is, we've got both like New World and Old World forms, just like the Trypanosomes. We've got Old World, New World type thing. What is also interesting about this group is the pleomorphy that it exhibits. Pleomorphy is simply saying that it has multiple body forms in their life cycle. So think back to our trematodes, to our cestodes and stuff. When we had development to very distinct life cycle stages, where the Myristidia looked completely different from the Rhedia, that looked completely different from the Cercaria, that looked different from the Metacercaria, and so forth. All right, you have those different forms, but they're very distinct. With the multiple body forms here, the pleomorphy, it's more of transitions. Yes, they're going to be distinct. Yes, I will ask you, what form is this? And you need to be able to identify it and know why it's that form. But it's more of a gradual. It's not like a wholesale complete shift in the body plan. So what are the forms? We've got amastigotes, promastigotes, opisthmastigotes, choanomastigotes, epimastigotes, and trypamastigotes. There's only 4 that I'm going to ask you to know. The amastigote, the promastigote, epimastigote, and trypomastigote. Why are those? Because those are going to be the 4 that we encounter in our life cycles, in our example life cycles. Yeah, opisthmastigote and choanomastigotes appear in other life cycles, but we're not going to talk about those species in this class.
So you said just 4?
Just 4 of them, yeah. I marked the ones with the asterisk that you do not need to know. So, what distinguishes these forms? Number 1, the flagella. Do we have a flagella or not? All right, the amastigote, so masti, it used to be called, be part of the Mastigophora, so whip-containing organisms. So masti would be the whip. With amasti, it's no whip, so no flagella. So that's what we have here for the amastigote. We have no flagella. All right, and then the other forms have a flagella. Next thing is the relationship of our kinetoplast and kinetosome to the nucleus. All right, so are we? I should arrange this. I have them set up so that anterior end is to the top, posterior end is to the bottom. As with most of our flagellates, when they swim with their flagella forward. It's a kind of corkscrew almost like. It whips around and almost makes it seem like they're going through a corkscrew. So we can talk about like where is it in relation to the nucleus. So number one here is all of our nuclei. All right, we have when our Kinetoplast can be in front of, anterior to the nucleus, antinuclear, or it could be post-nuclear, like it is down here. So trypanosomatida form is the only one where our Kinetoplast is post-nuclear. It's posterior to the nucleus. All right. And Kinetoplast is associated with the kinetosome, so if the Kinetoplast is posterior to the nucleus, so is the kinetosome. The other thing is a membrane, an undulating membrane. All right, so in these two stages, we have the flagella arising in a pocket, and then it extends the length of the body, but on that length of the body, you have this thin membrane, the thin cell membrane, that extends from the body and then encapsulates the flagella, the axoneme and the paraxial rod. So if we were looking at it, if we took this guy and cut him this way and then flipped it on end so that our flagella's at the top, this is our body. That's the flagella, ballet. This part is our undulating membrane. And we can see it in a few of our slides. So Trypanosomatida, our flagellar pockets at the extreme posterior. Flagella runs the length of the worm, or not of the worm, of our Trypanosomatida, our body, and it has this undulating membrane, almost like a ribbon-like membrane, all the way to the end, and then you have your free flagella at the very anterior end. When we go to the epimastigote, we still have that undulating membrane, but now our Kinetoplast is antinuclear, it's anterior to our nucleus. Still have flagella, still have the undulating membrane. Once we switch to a promastigote, now we are at our flagellar pockets at the most anterior end. There is no flagellar membrane at that point. And then a mastigote, no flagella, no membrane, so forth. Is there a flagellar pocket? Yes, flagellar pocket exists. And the amastigote still has a flagella, right? Because we have a flagellar pocket. It's still there, it just doesn't extend outside of the body. That's why we call it the amastigote. All right, we can see the nucleus, we can see the flagella, we can see the kinetoplast, and undulating membrane on our slides in the lab. Those are the things that we could see. Ready? This is our pleomorphy. And as we talk about transitions, this is one extreme, the amastigote's the other extreme. As we transition through life forms, we're going through the intermediate one. So if we go from a trypanosomatic to an amastigote, we're having transition up to there. We're going to have to pass from an epimastigote to a promastigote to an amastigote at that point. And you can kind of get an idea of what's happening. Is that you're getting the kinetoplast being moved forward, pushed forward. That gets anterior to the nucleus, and it continually moves to the anterior end. We'll see this in our life cycles. All right, so this was the order, right? Order Trypanosomatida. We're going to start in the genus Trypanosoma, and we've got two. We've got the African Trypanosoma, so the Trypanosomes from Africa, and then we're going to have the Trypanosome in America. So we're going to start with our African Tryps. So almost all species are heteroxynous, meaning more than one host. Our life cycle usually includes a vertebrate and then some blood-feeding insect, but we do have some where leeches are the vector. All right, so in the vertebrates, we find members of the trypanosoma in the blood and tissue fluid. They will be extracellular. In the invertebrate vector, invertebrate host, they're found in the gut, digestive fluids, the gut. When we get into the vertebrate, we can have a massive population size in there because we will see reproduction. You might have a single parasite individual get into that vertebrate, and then in hours you're going to start seeing millions of them. So we give example, 100 hours post-infection could have anywhere from 0.02 up to 4 billion parasites in the blood and tissue fluid. Do you think this is causing a pretty significant immune response? Yeah, it could. It could. All right. So these African trips have pretty big economic impact. The economic impact is somewhat on our agriculture, on ranching, but it's also could be on health. So with the health, we can think about lost worker hours. You know, how many hours are lost because of disease? Why is it so large? It's because the risk area of getting these guys exceeds the land area of the United States. Huge, most parts of Africa, you have a risk of getting this. You also have large parts of Africa where you can't raise cattle. Why is that? Because there's a species, Trypanosoma brucei, that will infect the cattle and cause this, which is Nagana. It's a withering disease of the cattle. No matter what they do, you can't get around it. And cattle are great, even though they are demonized. Right? They're important players. Ungulates are important recyclers in the community, in an ecosystem. Right? They eat vegetation, they break down the vegetation, return nutrients to soils. They do it much more efficiently than having it in a compost pile. So rapid turnover, and because they're doing that, you're basically taking this energy, producing it to meat, which then feeds individuals. But in these areas, you can't do that, which is why some of these areas, they still remain economically depressed. They can't get the cattle going until we can eliminate these diseases. All right, so Trypanosoma brucei causes Nagana. In our genus Trypanosoma, we have two different routes of transmission. And this is what distinguishes one of the key differences between our African trips and our American trips. All right, so with these transmission, it depends on where does our infective stage develop inside of our vector, inside of our insect host, and then how does that infective stage get to the vertebrate host? So I have a cross-section of an insect, actually a transverse section of an insect. So we can look at digestive gland. This is just kind of representative of a generic insect. But you would see your mouth, your pharynx, esophagus, basically your stomach area, proventriculus, your ventriculus here. You can have ceca, gastric ceca, your malpighian tubules, right, for water reuptake, so forth. Your colon, rectum, if we go by those terms. But instead of kind of giving all of the names, we can think of it as having a foregut, which is more of the head, a thorax region, your midgut, which is in the middle, and then the hindgut at the posterior part of the worm. So we'll kind of break it down: fore, mid, and hindgut. in our life cycles. So the first type of transmission is salivarian transmission. So based on the name, you should think immediately saliva. So it's going to be transmitted when the insect bites and delivers the infective stage via its saliva. This is also called anterior station development. This is kind of an older term to describe where this develops inside of our insects. So where does it develop? It develops in the anterior part of the digestive system. So for us, it's going to be the salivary glands or the mouth part. So you're going to have some development happening, some reproduction happening. Could be in the midgut, could be in the hindgut. But those stages are not infective to our host. Our parasite will have to move up to the anterior station, to the mouth, to the salivary glands. And then they will transform to a different life cycle stage, different body form. And once they complete that transmission, or that transformation, they are now infective to the host. This is what we observe in our African trypanosomes. You're going to get infected when they bite you and deliver it with through their salivary gland. Ready? All right. The American trypanosome, so Trypanosoma cruzi, exhibits circularian transmission. It's also called posterior station development. So this is kind of giving you, I don't know circularian, I don't know the root. I guess I should look up the root of circularian. Root of oh, hold on, here it is. Aha! Stirco, stirk. It's a new window. Check that out. Latin stircus, stircorus, dung, excrement, manure, feces, fecal. There we go. Know the root. Know what we're talking about. So we are in posterior. Now we have the same type of replication and stuff happening in midgut, possibly the hindgut. But those stages are also not infective to the host. They have to go into the rectal area or into the hindgut, transform to another body form, in which case they now become infective. All right, so we're getting development to infectivity in our hindgut portion. How is it transmitted? Well, it's not in the saliva. All right, so it's not going to be delivered when the blood-feeding insect feeds. Instead, it's going to be transmitted via fecal contamination of the bite wound. So we'll call it a bite and crap type of transmission, because that's kind of what happens when we have these rejuvids feeding. They start to feed on the blood, their gut fills with blood, their body expands, forces out anything that they have in the rectum. And that includes our transmission stages. Now, if it just stayed on your skin, nothing will happen. But we know that's not going to actually be the case, because if you get bit by one of these insects, your first response, if you're awake, is to smash it, in which case smashing it will release those infective stages, and you're basically rubbing them into the wounds. Or if you are asleep, you get bit, you generally kind of wipe them off, brush them off, but then you could scratch, and at that point you could scratch and move the infective stages into the wound. Hence the bite and craft. You can get it through accidental ingestion of the vector, so you're probably not eating these things consciously, But if you are asleep and you sleep with your mouth open, one of these things could crawl in and you could have a midnight snack and get get this parasite. They go into the mucous membranes is how they would enter your body. Or accidental ingestion of the vector's feces. So again, scratch, you get it under your nails, and then you bite your nails and you get it accidentally. All right, so salivary and circularian transmission, they distinguish between the American and the African trips. These terms only apply to the species of Trypanosoma. Leishmania no longer applies. All right? That's why I put it in caps. I was yelling at you through the screen. Only Trypanosoma. All right? All right. So we did our background. We're dealing with the African trips, so salivary transmission. There are two species that infect humans. Trypanosoma brucei gambiense, Trypanosoma brucei rhodesiense. How do you think they got their subspecies name? Based on the region. Where's our first one? Gambiense. Gambia, exactly. Yeah, Gambia. That is West Central Africa. West Central Africa. And then what about Rhodesiense? Rhodesia, right? That's more of a Central and East Africa. Right. So yeah, they do kind of separate. They are associated with different tsetse fly hosts, different species. So it's somewhat host dependent. That's kind of what triggers their ranges, and their hosts have certain environmental requirements. So if we go farther west, you're talking about drier areas. When you go more central and east, you have wetter areas. That's kind of where the vector can live and reproduce. All right. So they use different hosts. They did different locations. Also, different type of infection. So Gambiense is probably the less serious. I mean, both of them are serious, but Gambiense is probably the less serious. It can develop a chronic form of trypanosomiasis, which would be our African sleeping sickness. Rhodesiense is more of an acute type of infection. Doesn't normally get to the chronic stage because the host is normally killed before it ever gets to that stage. That's why we say it's more of an acute. Both of them, again, will cause pathology. If left untreated, both of them can't kill you, but definitely Rhodesiense normally will take you out in about a month or so, if left untreated. There is a species of brucei that is important to cattle, and we mentioned that. It's Trypanosoma brucei. That causes Nagana. That's the one that basically has prohibited communities from raising cattle. Ready? Africa's a big continent. Big continent. All right, let's go through the life cycle. Right in the middle. Let's grab a piece of chalk. We'll do the life cycle and we'll pass out the exams. I'm going to say this is a fairly simple life cycle. We do have a couple that are somewhat difficult, somewhat long, somewhat challenging. This one is not one of them. So, this is Trypanosoma brucei. So fortunately, those three species that we have, Gamiense, Rhodesiense, and Brucei, three subspecies, they all have the same life cycle. So once you learn it once, you know it forever. All right, so our host, our vector is the tsetse fly. And then our definitive host, or our vertebrate host, is the mammal. And we'll kind of get off of the definitive intermediate host because it's going to be difficult to kind of say where the sexual reproduction occurs. Some of it, like this one, it's all asexual reproduction. So we have our mammal host, and then we have our insect vector. So we're going to start in the tsetse fly. We have a metacyclic Trypanosomatida. Metacyclic Trypanosomatida. We'll give a location. It's in the salivary gland, but I'll add that as we get to that. So we are in the salivary gland, and this thing is, tsetse fly is going to bite. Blood meal. And deliver our metacyclic Trypanosomatida to our mammal. So the Trypanosomatida is our body form. Metacyclic is just a term that says we are infective. All right, this is our infective Trypanosomatida stage. It goes into our mammal, so blood meal delivers it. It's injected basically. We're going to have a Trypanosomatida in blood, could be the lymph, and tissue fluid. So it's going to occur, all of these stages occur extracellular in the blood, lymph, or tissue fluid. So we're going to have our Trypanosomatida, and it's going to be what we call the slender form of our Trypanosomatida, which is long and thin. This stage will replicate via binary fission. Technically longitudinal fission. I mean, longitudinal fission is binary fission, but it's on the longitudinal axis. All right, then if we look at our you could say that we are a long, slender Trypanosomatida. Replicates. And then eventually, it reaches a population size where some of them will start to transform. It goes through an intermediate form and then transforms to a stumpy form. Still a Trypanosomatida. So our stumpy form Kinetoplasta, nucleus, bigger and thicker. I should say thicker. Slender, thin versus thick. This is a stage that gets picked up by our insect during a blood meal. So this is all extracellular on that side. Blood meal ingests the stumpy form of the Trypanosomatida, and then it's in the midgut, and it is now a procyclic Trypanosomatida. Procyclic Trypanosomatida just means that we are replicating. It's going to do this for about 10 days. going to replicate. Oh, and this is in the midgut. It's in the midgut. And then what's going to happen is some of these will migrate to the salivary gland and transform to an epimastogote stage. So with our epimastogote, remember, our kinetoplast becomes anterior to our nucleus. This is in the lumen. Of our salivary gland. All right, so it transforms to the epimastogote, and it's going to replicate just for a few generations. It's going to replicate a few times, and then they will attach to the villi of the salivary gland. So they're going to attach the epithelial villi of the salivary gland. They're going to attach using, I believe, their flagella. They attach. And then they undergo a transformation to our trypanosomatic stage. And once they complete transformation, they then detach. And then they detach. And once they detach, they're where they need to be. Metacyclic trypanosomatic in the lumen. So when our trypanosomatic, or when our insect feeds, it can inject these stages in the blood. How long does this take? Once we take that blood meal of our trypanosomatic stage, it takes about 3 to 4 weeks for development to our infective stage. Questions? It's not terrible, fairly simple. Fairly simple life cycle. But a few things. We are all extracellular. We have a transition to a stumpy form. We get over here, we're going to be in the midgut as a trypanosomatic, replicate asexually. And then we go up to the salivary gland, where we then transform to an epimastogote stage, undergo a few rounds of replication, attach to the salivary gland itself, to the cells, and then transform to our trypanosomatic stage and detach. Why the transformations? We'll talk about that on Monday. All right. So review this. I'll pass back the exams.